Before we go on to that, though, I wanted to briefly discuss some interesting results around the current COVID pandemic. As many of you know, COVID has a variety of symptoms, and one symptom that isn't exactly uncommon. In fact, it appears to be a relatively common symptom of SARS-CoV-2 infection is loss of smell, which is known as anosmia. So in individuals that are infected, they have been reporting that they just simply can't smell. And in fact, that this symptom can persist for months. Now, superficially, this would suggest that the virus is perhaps infecting the nervous system. And you could come up with one reasonable mechanism, which might be that perhaps you breathe in the virus. You know that, as we learned in the previous lecture, that we have olfactory receptor neurons that have these cilia that are really very close to the nasal cavity. You could imagine that that might be a route for the virus to then get into the nervous system. But in fact, it turns out that neurons throughout the central nervous system and even the peripheral nervous system, they lack the expression of the proteins that are necessary for SARS-CoV-2 that it uses to infect those cells. So this is a figure taken from a paper that was recently published in Cell, kind of collected a bunch of results on anosmia, and what's been most recently been focused on are some studies that have looked to see which cells are expressing these important proteins, known as the ACE2 receptor and then TMPRSS2. So apparently these two proteins are needed for SARS-CoV-2 to infect cells. And so we've got three different layers in which we can look at within the smelling abilities. If we look at the olfactory bulb, they've got a little schematic diagram of the variety of cells there. This is showing more cells than what we talked about in the smell lecture, but it does include some of them, right? So we've got glomeruli, and that would include the afferents from the olfactory receptor neurons, and then the efferents from mitral cells. There are also other cells in there called touch cells. And then we also have these granule cells that we discussed as well that make up the center of the olfactory bulb. And if you look over here, they've got kind of a grayed out schematic of which cells in the olfactory bulb are expressing ACE2 and this TMPRSS2. So the ACE2 receptor, this is the thing that I know that the little spikes on the SARS-CoV-2 virus, that's what it attaches to. And it looks like there's only a single cell in the olfactory bulb that expresses this combination of proteins, and they're called pericytes. And so these pericytes, they don't actually have interaction. There's nothing about the pericytes that are necessarily connecting directly to the nasal cavity. So it kind of rules out that this part of the olfactory system is going to be compromised directly by SARS-CoV-2 infection. But when you look at the olfactory epithelium, which is innervated or innervates the olfactory bulb, I should say, we discussed cell types in the olfactory epithelium. So here in red are the olfactory receptor neurons, and then we have these supporting cells. They're called sustenticular cells, and there are other types of cells, microvillar cells. And then also importantly, there are these horizontal basal cells, which divide and give rise to new olfactory receptor neurons. These basal cells and the supporting cells in brown, they express both of these receptors. And it looks like that these are the cells that get infected with SARS-CoV-2, and they probably become inflamed and die. And so if these cells are dying, what's likely is happening over time is that you have a compromised olfactory epithelium function. So the supporting cells are no longer there to provide mucus, to allow for the cilia to do their job. They do other functions such as sort of maintaining ionic balance within the olfactory epithelium. If you have those cells being inflamed, or even if they're dying, you're going to have disrupted ionic balance, and that will affect the the olfactory receptor neurons function. And then the basal cells, if they are dying, then you're going to lose those basal cells that would be then proliferating and giving rise to new olfactory receptor neurons. So, so these are all things that clearly could have an impact on smell olfactory function, and certainly that's got to be what's happening with anosmia, is that it's olfactory epithelium that's being infected. So, little side note that's really interesting and obviously very relevant to the whole SARS-CoV-2 COVID-19 pandemic. And yeah, I'll try to remember to bring these things up as we go along through the semester, because COVID-19 actually has an impact on behavior and cognition in all kinds of ways. And we will touch on those things in the future. So I wanted to kind of stop and get a brief overview of where we've been so far in thinking about the sensory pathways in the brain. We have so far talked about vision, and these are the receptive cells associated with vision. We've got the rods and cones. These are the cells that translate physical information in the world into the information that can be understood by the nervous system. We also have talked about the chemical senses, smell and taste, and we just discussed those, including the olfactory receptor neurons, the green cell on the left. And so we still have two other major categories to discuss, and for the next two lectures we're going to be discussing touch, and we will be talking about the specialized cells that are needed for taking in physical world information and translating it into the information that the nervous system can understand. So when we refer to the touch pathways, these are known as somatosensory pathways. This includes perception of touch, pain, proprioception. There's also some chemical information that gets translated through somatosensory pathways as well, related to pain. We will be walking through the various steps in translating this information. I want to review these steps again, right? That sensory pathways and somatosensory pathways are not any different. The first step is sensory reception. You have receptors in the periphery that detect a stimulus. And then there is sensory transduction. In response to this physical stimulus, the receptors, and it may be ion channels, typically that's the case, but it isn't always. Sometimes it's G protein coupled receptors. They may open and close. This will, but eventually that leads to a change in neuronal membrane potential, right? So we have sensory reception leading to transduction, some change happening within that receptor cell. Then, once we have that change in neuronal membrane potential, then there's transmission happening, where an action potential at some point is propagated within the periphery, leading into the central nervous system. Eventually, we have perception. The brain has to receive the sensory information, process it, and then the organism becomes aware of the stimulus. So keep these four steps in mind as we talk about somatosensory perception, and in particular, we're talking about touch and proprioception. So, what is somatosensation? Somato actually means body. So in fact, soma, we talk about neuronal soma, those somas are the cell bodies. Same word, same root. Somatosensation then is the sensing of changes within the body. Generally, this may be thought of as sense of touch, but as I've already mentioned, there are several different sensation types. We do, of course, pain is critical for understanding how somatosensation works. We also have temperature, something that we will be discussing. There is fine touch and coarse touch. Those are two subcategories of touch. Pressure, vibration, and then we include in this, because it uses a lot of the same pathways and even some of the same kinds of receptor mechanisms, body position, as I said, proprioception. So there are three main categories of somatosensation receptors that we're going to be talking about, and then within those categories there are subcategories, of course. So a lot of the somatosensation is conveyed through receptors that are just located in the skin. The exceptions to that are some of those receptors that are involved in proprioception. But basically everything else is in the skin. So these three main categories are mechanoreceptors. These receptors sense changes in pressure or movement. We also have the nociceptors, and these receptors respond to pain stimuli. And then we have thermoreceptors. These receptors detect changes in temperature. They can also detect certain chemical changes. Today, we are only going to be talking about mechanoreceptors. We will talk about the other two categories in the next lecture. There are 4 different types of skin in humans. And one of the coolest things about the nervous system is that it is, it's a 3 to 5 pound organ in your body. It takes up around 3% of your body weight, but it touches upon every single part of your body and interacts with virtually every single part of your body, including all across your skin. So your entire skin is innervated by the nervous system. And so we have to learn about, as neuroscientists, we don't just focus on, say, just the brain, like someone might say they focus on just the heart or the liver. The nervous system, it is fully integrated with all these other systems, so we have to learn stuff about those other systems. Skin, obviously, is no different. So we're going to talk about these different kinds of skin types. So the first one is mucous membranes. So this is the lining inside of body orifices, such as inside the anus and inside the mouth. There are the mucocutaneous types of skin, and that is a special type of skin that is at the junction of mucous membranes and then the other main types of skin that we understand. And so examples of those, of the mucocutaneous, is the lips, the nostrils, the vagina, foreskin, and the anus. So then there are 2 main kinds of skin that we are most familiar with. One is hairy, and that is, as you might have guessed, skin that has hair. And then there's glabrous skin, which is kind of a gross word to say, glabrous, kind of blah, out of your mouth. But that's skin without hair. And so that might be like your forehead or skin on your fingertips, that kind of thing. Yeah, so those are the main types of skin. And talking about some of the differences between them and how mechanosensation is different between them. Mechanoreceptors, we're going to discuss first the mechanoreceptors that process tactile information in glabrous skin. And we're covering this because glabrous skin is where we have some of the most sensitivity to touch information. And that's because that includes your palms of your hand and your fingertips. This is where you're going to experience fine touch, and so that's a good starting place. So fine touch is regulated by several different kinds of receptors. First, there are the Meissner corpuscles. These guys are located here. They're in the epidermis of the skin, and they're particularly sensitive to skin deformation and texture of objects moving across the skin. And this is really important for things like grip control, so holding some sort of tool in your hand and knowing the position of where it's at, and if there's like a little switch or something, your Meissner corpuscles are going to be involved with that. Then, in addition, there's Merkel cell afferents, and so these guys are located down here. These are sensitive to points and edges and curvature, so they process form and texture. So knowing if something is curved or if it has an edge to it, your Merkel cell afferents are going to be sensitive to that. So in addition, though, we also have, in deeper layers, Ruffini corpuscles, and these are located deep within skin inside the dermis, and they are also, you can find Ruffini corpuscles within ligaments and tendons. This makes them sensitive to cutaneous stretching, such as when you move your fingers around or when you move your limbs. These Ruffini corpuscles, it's not to say that they aren't activated through touch, but they tend to be more involved with proprioception. And so we'll revisit the Ruffini corpuscles in just a bit. And then last that we're going to discuss is the Pacinian corpuscles. And these guys are sensitive to vibration. They're known to be associated with like skilled tool use, such as like using wrenches or cutting bread with a knife, or you will use these as you're writing, so as you're taking your notes. The Pacinian corpuscles happen to be pretty heavily involved with this. So we're going to go through each of these receptors, and we're going to see how they become activated when we are processing tactile information. And one type of tactile information that is where especially it's dependent upon fine touch is reading Braille. And for those of you that aren't aware, Braille is a form of written communication that can be used by people who are blind or anyone who understands Braille. So it's a series of raised dots that will correspond to a letter in the alphabet. And if you put them in a sequence, it's like you're spelling out words. So, but the neat thing about Braille is that it's not a visual related form of written communication, it's tactile. And so you have, you're going to be using your somatosensation in order to read Braille. So what we're going to be looking at here is how these different afferents respond to Braille, so this fine touch perception. And so what we're looking at is essentially what we're seeing is how these cells respond, and the relative position of how they respond as the finger is drawn across the alphabet here. This is the alphabet. And what you can see is that the readout of the information that you can get from these particular mechanoreceptors varies, and it should give you a sense of which ones are providing the best fine motor or fine touch perception and which ones are not. So Merkel cells, you can see that they are sensitive to these points and edges and curvature, and that they can process form and texture. Clearly, you can see the way that they are responding, that they're very sensitive to these dots, and that this is going to be the information that's going to be most salient to the nervous system in reading Braille. But Meissner corpuscles also clearly carry some of this information as well. That you can still, it may not be necessarily as specific as the Merkel cells, but you can still get some readout from Meissner corpuscles. When you're looking at Ruffini corpuscles, you don't actually see much of a pattern. There's a little bit, but it's likely not that this is actually giving much information that the nervous system can understand. And the same goes for the Pacinian corpuscles, where even though reading Braille is going to be sort of a skilled activity, it is something that is dependent upon textures and fine touch, and the Pacinian corpuscles just simply don't translate that information, and you can see that there really isn't much of a pattern here that matches the finger as it's going across. Whereas with the Merkel cell, you can see you've got A here, and as it hits B, then you get the activity there, and As you go all the way down to the alphabet, you get 3 dots here and 1 next to it, and it's showing up quite well. So mechanoreception is going to have different levels of sensitivity depending upon whether you're doing something like skilled use or fine touch, but it's not like that there is only one single receptor that's only going to contribute to that particular activity. It is going to be the combination of the activity happening across the mechanoreceptors that the nervous system will use to read out and understand what's actually happening and what is being touched. Hairy skin has a very different set of mechanoreceptors than glabrous skin. In hairy skin, what we have fall into 2 basic categories. There is mechanosensation directly into the skin, but that is typically done through what are referred to as free nerve endings. And we will talk about how all of this is connected up to actual neurons, including the mechanoreceptors in glabrous skin in just a bit. But free nerve endings, they don't have any sort of particular shape. Instead, that these are the ends of nerves, of somatosensory nerves, that are just innervated into the epidermis and sort of branch out. And we'll cover how they induce active potentials in just a bit. Hairy skin obviously has hair, and you can sense things through hair. Some animals actually depend upon hair as a critical component for perception of the world. In particular, you can think of whiskers, and many mammals will use whiskers as one way that they perceive the world and go through mazes and tunnels and that kind of thing. Especially, small little rodent-like animals have very complex series of the whiskers, and the way that's all related in the brain is actually a super interesting topic that we're just unfortunately not going to be able to cover today. But all hair, including human hair, will have some kind of mechanosensation that occurs. So hair follicles, we have a couple of different kinds of hairs on our skin, but they all will be rooted in the dermis, and then you have nerves that will create these sort of rings of afferents that go around the hair follicle. And as the hair gets displaced, so as, say, a motion comes and pushes on the hair this way, they will press up against those afferents and induce active potentials that way. So the specific names here, I don't expect you to have memorized. You do need to know free nerve endings, but just being familiar with this basic mechanism of having these rings that go around the epidermis, around the hair follicles, that's the most that you would have to know about that. So those are the receptors. Now we got to talk about transduction. How does this touch information get relayed into the into information that the nervous system can actually understand? That's the sensory transduction part. So we've got a physical change happening. That's what mechanosensation is detecting. When you touch raised dots on Braille, there's physical change that's happening across the skin over time. Say something brushes against the hairs on your arm, there's physical change happening onto those particular receptors over time. So what is actually happening then within the cells? It's a pretty relatively simple mechanism. So in fact what happens is that whether you have some sort of corpuscle that is sort of complex, with like the Pacinian corpuscles tend to have this kind of overall morphology with layers and layers of membranes that cover up an encapsulated afferent fiber. Other kinds of receptors, like the Meissner receptors, they actually are a collection of the afferents that are sort of twisted upon each other. They all kind of work in a similar way in that a stimulus will come in and press upon the, say, membranes, or as far as free nerve endings go, press upon the free nerve endings and induce stretching along the membrane. And this physical stretching along the membrane of these cells will cause changes in sodium channels that are present there. And when the membrane is in the stretch conformation, or something is pressing upon that, you're going to have these receptors actually open, and you'll have influx of sodium channels. So influx of sodium going through these stretch sensitive sodium channels. So a little bit of stimulus, like a weak stimulus, something just pressing on it just a tiny little bit, you can induce a membrane potential change within the sensory afferent. So let's say we're recording from that afferent, but we're pressing just a tiny little bit. So that's what's happening here. You got it going up a little bit, tiny little bit of force, and then we turn it off. You can sense what's happening within the afferent. You can see a membrane change in membrane potential, but these are neurons, and they, just like the action potentials that you learned about in Neuro 1, they have to reach a certain threshold in order to have an action potential, right? So a weak stimulus is going to be, in this particular case, it's going to be below the threshold that's needed to induce an action potential. We increase the stimulus a little bit more, we're getting closer, but not quite. But eventually you'll reach a stimulus that will induce sufficient changes in membrane potential that it reaches the threshold for inducing action potential within that cell. And so then you will actually get a spike or several spikes that are happening depending upon the stimulus. So afferent fibers that are encapsulated by these kinds of receptors, like the pacinial corpuscles, or having other things that are sort of covering the free nerve ending. So all the four that we talked about in the glabrous skin, these are referred to as mechanoreceptors because they have this sort of complex ending that's covering the nerve ending. They're not free and open. But fibers that lack these kinds of receptors, these are referred to as free nerve endings. Pretty simple name. So a very, very similar mechanism in what's happening electrophysiologically, but by adding these different kinds of encapsulations onto the ends, that's where you can get the differences in touch sensitivity that allows for fine touch or vibration or that kind of thing. So now we need to talk about receptive field size. We already discussed this a fair amount with the visual system, where different groups of retinal ganglion cells will have a receptive field that they have. And we talked about how the receptive field size is much larger in the periphery of the retina than it is towards the fovea. If we look at the fovea, so these green, that refers to the cones, you can see that the receptive field size for retinal ganglion cells that are attached to the cones that you find in the fovea, the receptive field size is very, very small. But as you get farther and farther away from the center point of the focus of our retina, the receptive field size actually gets larger and larger because we don't need that fine degree of sensitivity in the periphery. It's good enough for us to pick up some color information and light information and movement, but we don't need sharpness of the image in order to have good visual acuity at the periphery. We just need it in the center of our vision. So that means that we have high sensitivity, small receptive field size in the fovea, and relatively low sensitivity and very large receptive field size in the outer parts of the retina. Skin has something similar to it. We have receptive fields on our skin. These receptive fields, we refer to them as far as the skin goes, the area of the skin where the stimulation alters firing of a sensory neuron that's innervating that region. So if we wanted to go and measure the activity of a particular neuron, we could measure out precisely what its receptive field is. And there's a really simple way that we go about doing this. You can take a set of calipers Google it if you don't know what I mean, what calipers are, but calipers will be, it's a device that has 2 points at the end of it, and you can use it to measure the distance between those 2 points. And you can use these calipers as well to measure the overall receptive field size. So one way that you would go about doing this, and I think that you guys do this in lab, unfortunately it involves touching, so hopefully, I guess you have to do it for yourself. That's okay, right? But this is it's a very common laboratory exercise to understand how receptor field size works. You take these calipers and you touch it onto your skin at really small distance, and if it's at a really, really small distance, you're likely to only perceive it as just a single point, even though there may be 2 points with a tiny little bit of distance between them. But then you start increasing the distance of between the 2 points more and more until you can perceive 2 points. And that threshold at which you perceive 2 points, that is considered, that will be one way of measuring the receptive field size. Certain body regions have larger or smaller receptive field sizes. As you probably know, that because we have fine touch at the glabrous skin, and we have all of these different receptors that are within glabrous skin, our receptive field size on our fingertips is much, much smaller than it is, say, on your wrist, which is what we're seeing here. And they're showing like how you can differentiate the receptive field size here. So this is, say, the receptive field of one particular receptor, and here's the receptive field of another particular receptor, and you would need to eventually reach that distance between the two where there's no overlap between them so that you can perceive the distance. And you can see the distance between those two receptors is actually quite a bit bigger than the distance on your finger. Your finger has the greatest level of sensitivity. Actually, I think the lips are slightly smaller receptive field sizes than the fingertips. But yeah, that's where you're going to find the smallest receptive fields with the highest level of discrimination capabilities at the fingertips. The receptive fields, this is showing what they look like, both the left and the right, on this individual here. It's not wearing any clothes. Okay. So the size of this activity where you're using the calipers, this is known as the mean 2-point discrimination threshold. So what would be the average for those 2 points that you can find on any of these parts of the skin? So as you can see, some of the smallest parts are the hands and the fingers. Also the lips, this is showing it's slightly higher. I think I've seen data where it's actually smaller than the fingers, but whatever, they're pretty close. And then the face is also has some of the smallest 2-point discrimination thresholds. But areas where we really don't need to be able to discriminate fine distances between two objects are things like the thighs and the calves and your arms, your back, and your belly. You're not really doing like a whole lot with those areas that would require fine discrimination of 2 points. It is going to be your hands, and then there's a lot of important things going on at the face. There's kissing, but it's also like it's got a bunch of your critical sensory organs for seeing and smell, and so you need to have good sensitivity there so that you don't lose an eye or have something crawl up your nose. So that's why there would be that level of sensitivity that's necessary. It's really just like the hands, the face, and the feet where you're going to have some of this better sensitivity. Everything else is going to have relatively low sensitivity. Very large receptive field sizes. All right, so that's about the periphery. Now we're going to start moving into the central nervous system here. This on the left is a really kind of disgusting image. Took off the internet someplace. Showing in humans the basic anatomy of what's going on inside the body as far as how the spinal cord is organized. So this is going to be one of the first places where we're really talking about the spinal cord and how it interacts with the rest of the body. And so I want to spend a little bit of time talking about this anatomy because we're going to be talking about spinal cord anatomy quite a bit in this class. So this is maybe disgusting, but it's a good overview of what this looks like. So I'm going to refer to some of these these parts, and I think this is the only time I'm going to show this image, but I think it's kind of important that you see what it looks like. So this is in this individual, what we have is the we've got number one is the spinal cord, right here in the middle. And then number two is something that's very important that I want to spend a lot of time talking about, which is called the dorsal root ganglion. So these are very important for mechanical sensation. Okay, so there are these in your body along your spinal cord. You have these nodules. They're like bumps within the different nerves that go into the spinal cord. And these bumps are referred to as the dorsal side as the dorsal root ganglion. Let's see. Number three is rootlets of spinal nerves. So the spinal nerves, they sort of, as they come into the spinal cord and go through the dura that's on the outer part of the spinal cord, the nerves are they sort of branch out into different places and innervate in sections along the spinal cord. What else is here? Number five is a spinal nerve. Yeah, and number six, as I said, that's the dura. So the dura that surrounds the brain also extends down into the spinal cord and protects the spinal cord as well. So yeah, that's what that looks like. Now if we go over here, what we're looking at here is a cross section of the spinal cord, and we're going to spend a little bit of time talking about the basic anatomy of the spinal cord. So this is a simplified schematic of how this all is wired up, and we'll be revisiting this again also in the next lecture when we talk about pain and temperature. So we're not going to talk about the blue part, but instead today what we're focused on is the red part. So tactile sensation, it begins in the afferent fibers with the receptor endings in the skin or the muscle. So here you might have receptor endings that are carrying Pacinian corpuscle information, for instance, that you would find in the glabrous skin at the fingertip. This information, so you're inducing, so at the scale that we're looking at, I can't even see the receptors there, but let's say it was sufficient enough to induce an action potential, then the action potential is going to propagate down this fiber, and it's going to pass this and pass through the dorsal root ganglion. And so this is kind of important. So these bumps here, this is where the cell bodies of where these cells, these sensory afferents are located. So the cell bodies are located right here in the dorsal root ganglion. So this is the reason why you have these bumps, because this is going to be loaded with not just the two fibers that we're looking at here, but all the fibers that would be innervating, say, this part of the skin. And so you'll have this big old bump that's filled with the cell bodies of all those afferents. Okay. So this is the dorsal root ganglion. Now the rest of the sensory neuron, then, as it will pass through here at the action potential, and then it will go into the dorsal side here in what's called the dorsal horn. And then we'll talk about where it goes from there in just a bit. These sensory neurons, we refer to them as pseudo-unipolar neurons. So they're called pseudo-unipolar because they basically have this one long process, but it's pseudo because they're actually sort of broken in half right there. Yes, to repeat what I just said here, we have the peripheral and the central components. Here's the peripheral, central, and it looks like one long fiber, but you've got the cell body here in the middle, but it's kind of branched off. And so the action potential will propagate along this fiber, and not necessarily involving the cell body in the action potential itself. The cell body is there to keep the cell alive and do all the other things that cell bodies need to do, such as store the DNA and make stuff. So that's the overview for this. If we look at this diagram I just threw here over on the left-hand side, what you can see, and I hope that you can appreciate, is that now we're sort of bringing up the scale a little bit. So you have your finger here. Take this finger, it's over here. The actual receptor is located here, but this is the sensory receptor neuron. It will then go through a particular dorsal root ganglion and be connected at that particular dorsal root ganglion. So this particular receptor is located there, but there's going to be a bunch of receptors that will still pass through the same dorsal root ganglion and then go up into the brain that way. And the other thing that I like about this figure is that it shows how this is all organized, right? You've got this line of nerves that are coming into the spinal cord from the skin, and you can see that it's almost like a bunch of grapes on a vine or something like that. I don't know. But it's a bunch of little, all these bulbs right there. Those are all the dorsal root ganglia. And the thing that you need to keep in mind is that all of those dorsal root ganglia, and it's organized in this fashion, is because our body actually has segments all along the body, which is kind of crazy. We refer to these segments as dermatomes. So these are going to be the territory of the body that is innervated by a single dorsal root ganglion. And so there's an organization that happens to this. It's not that a single dorsal root ganglion will just kind of go all over the place. It goes into the body in a very organized way, just like our retina goes into the innervates the lateral genicular nucleus in an organized way, and in order to maintain the retinotopic information, where on the retina is light hitting, we got to keep that information organized within the central nervous system as we go farther and farther back. Somatosensory information is the same way. There's going to be a map that is going to be critical for how this information is understood by the central nervous system. The eyes work in the same way. They have a map. So does the skin. So and we refer to this as a dermatome. So this is going to be a particular area that's innervated by through one single dorsal root ganglion. And so this is showing along the skin where you can kind of map these things out. The spinal cord is broken up into 4 basic parts. You have the cervical, thoracic, lumbar, and sacral portions of the spinal cord. And then this is the side view of the same thing. The nerves that come out of those areas, they innervate particular lines along the skin. And so let's say there was damage to one particular nerve, or say a dorsal root ganglion was severed or something like that, that individual would lose then sensation along that particular dermatome. So physicians can use that information to try to narrow down where damage is located within, say, if someone is coming and presenting with some loss of sensation along the skin, say through a spinal cord injury or some other injury that's adjacent to the spinal cord, you can sort of figure out where that damage might be located by just simply assessing where they still have sensitivity along the skin. And you can figure that out. And so that by just using this map. Yeah, so that's dermatomes. So in fact, spinal cord trauma is a major issue. And this is an important point to talk about this, and we will also revisit it when we talk about the motor output. But when you have a spinal cord transection at a particular part of the vertebral column, you will then have total sensory loss of everything below that part of the spinal cord. So if, say, an individual has a transection of the spinal cord at T1, which is the top portion of the thoracic cord, they will lose all sensation within the thoracic, lumbar, and sacral regions. So they will have sensation along their arms, but nothing below that. Nothing in the thorax or in their legs. Let's say an individual just kind of has a spinal cord transection that happens within the sacral portion of the region. that person will only lose sensation in some parts of the limbs and then along the genitals, because that is the portion of the spinal cord that innervates genitals and like along the anus. So an injury along the spinal cord will determine the severity of the actual injury. So the higher up it is, the worse it will be for that individual. So damage to the sacral portion of the spinal cord, it can have serious consequences, but it's not nearly as debilitating as a severing of the spinal cord in the cervical region, where then you will lose not only sensation of the lower limbs but also the upper limbs. When that happens, when you've lost all sensation of the upper limbs and the lower limbs, that's called quadriplegia. And then injury between T1 and L1, so if the injury occurs in here and you lose all sensation to the lower limbs, your legs, that's known as paraplegia. So you're not going to have loss of sensation to the arms and still maintaining sensation to the legs. Those are the only two forms. You either lose just the legs or you lose both. Common causes for spinal cord injuries are generally from accidents, and the most common is motor vehicle accidents. That's where you're going to have enough serious force applied to the body that'll lead to the severing of the spinal cord. Sports injuries, though, can result in spinal cord injuries. Famously, Christopher Reeve, who played Superman. He's most famous for his role as Superman in the 70s and 80s. This was after he had played those roles. He was an avid horseback rider and was in, I think, a competition when this actually happened. He fell from his horse, landed on his head, and had a cervical dislocation, and he lost all control and sensation along his arms and his legs. And he was a huge advocate for research into spinal cord trauma, and a lot, still even a lot of research and work is to the benefit of the Christopher Reeve Foundation. So that's spinal cord injury. It is something that we will also revisit when we talk about motor systems too. There are different types of afferent fibers that carry information to the central nervous system. And some of the most clear and obvious differences is just the size of the fibers. We'll be looking at this again in just a bit, but we'll be revisiting this in the next lecture when we talk about pain and temperature information. But for the main two that we're talking about today, which is proprioception and touch, these fibers tend to be quite large. So the afferent fibers, neurons can actually vary quite a bit in the overall size of the fibers. So you can see the axon diameter for those involved with proprioception, which is our sensation of our position of our body, is actually up to 20 micrometers in diameter. It's quite a large fiber. Touch can be upwards of about 12 microns in diameter, which again is actually still quite large. Many axons and fibers within the central nervous system, they're not going to be any larger than a couple of microns. But when you're talking about proprioception and touch, those fibers are actually quite large. The larger the fiber, the faster the conduction velocity. So you can see the conduction velocity for proprioception is on the order of 20 to 120 meters per second. Keep that in mind. So around, on average, 100 meters per second. That is essentially the length of a football field in 1 second. And then if you compare it to pain and temperature, that's traveling at 1/2 to 2 meters per second. So there's a huge difference in overall conduction velocity that can happen here. The other thing that you should keep that should be obvious from this is that proprioception and touch, these are myelinated axons, and so those are things that will obviously contribute to having a faster conduction velocity. And I know in Neuro 1 We learned about myelination, so we're not going to talk about that. But it is critical that you know that proprioception and touch, those fibers have are myelinated. The fibers that we're talking about today, they're known as 1A or 2 or A beta fibers. Okay, so 1A or 2, that's the proprioception, and then A beta are the fibers that we're talking about today. Okay, so that's touch. That's getting up to the spinal cord. We still need to talk about proprioception, and so I wanted to cover that here. Proprioception is as I said before, it's the perception of movement and position of the limbs and the bodies in space. And they use certain kinds of mechanoreceptors that are located within the musculoskeletal system. And these receptors, there are 3 kinds of receptors that we're going to talk about, which is muscle spindles, Golgi tendon organs, and joint receptors. And this is the kind of perception that tells you whether you're sitting, standing, whether you're kind of standing on your arms and your legs, and would be very important for knowing whether you have to have your left arm on the green dot and right arm on the yellow dot and that kind of thing. Muscle spindles, they're found in nearly all skeletal muscles, and they detect muscle length and position. This is an example of what a muscle spindle looks like. So within a particular muscle, you will have what are very similar to the Ruffinian corpuscles, that's kind of this oblong shape, and then has the ability for stretch. So when a muscle stretches, there'll be tension that will happen onto the fibers, and that will induce a change in the overall membrane stretchiness, and will act upon those mechanically gated ion channels in the nerve endings. And that can induce, if the stretch is enough, that'll induce an action potential, and then send the signal on up into the brain. And as I said, these kinds of fibers, they use group 1A and group 2 fibers, which are the largest and fastest that you're going to find. And they're both myelinated. There's some differences that are carried between these 2 afferents. So the group 1A afferents, they're very large myelinated sensory axons that transmit information about limb dynamics, such as velocity and direction. So these are things that can be picked up by these kinds of fiber connections. And then the group 2 afferents transmit information about the static position of limbs. So if your knees are bent or if they're straight, it'll be group 2 afferents that'll carry that information. The density will actually vary quite a bit. So large muscles that are involved with coarse movements, like your calf muscles, they're going to have fewer spindles, because you just don't necessarily need to have that, again, sort of like the receptors within the skin. You just don't need to have that fine level of sensitivity for having that level of information. But the muscles in your hands and your neck, the muscles that control eye movement, there are tons of spindles in those tiny little muscles. And so the receptive field generally then is going to be much smaller, so there's greater sensitivity, because position is going to be so critical for those kinds of muscles and for the things that they do. There's also something called a Golgi tendon organ. These are located in the tendons, and they will tell you something about muscle tension. So if a muscle is pulling on a tendon, what you will find is that then that's going to stretch this network of collagen fibers that will have the nerve endings of an afferent sort of mixed within those fibers. And so then the fibers will kind of press on and stretch the collagen fibrils will stretch the free nerve endings, causing mechanical sensation to change, and then that will induce an action potential in the afferent. These joint receptors, they're present around the joints, And they also provide additional information about limb position and movement. Again, very similar to the information carried by by fibers that are within the muscle spindles. So those are going to be the two main types of receptors that we're going to talk about for proprioception, and that's going to be it primarily for proprioception. Now we're going to talk about how this information gets into the central nervous system. So that's going to be the transmission part for this. And so I wanted to walk through those steps. Mechanosensation, proprioception are carried up through the brain, touching upon, we're going to talk about 4 neurons that are in a series. Obviously, there are millions and millions of neurons that are all running in parallel, but we're going to schematize this by drawing out single neurons. So this pathway, like I said, consists of 4 neurons in a series. The first, obviously, is the one that we've been spending the most of the time talking about, which is this primary sensory neuron that's in the periphery. Its cell body is found in the dorsal root ganglion. It has either a mechanoreceptor or, say, free nerve ending out in the periphery, or, say, maybe it's coming from one of the joint receptors as well. They all pass through the dorsal horn and then carry up to the medulla. So they go up to the medulla, and they connect to the dorsal column nuclei of the medulla. So this is the dorsal side of the medulla, and this is called the dorsal column nuclei. So they synapse onto those neurons in the medulla. So keep in mind, these neurons are some of the longest neurons that you're going to find in the entire body. This could be a receptor down at your big toe. It'll be carried up to the sacral dorsal root ganglia, which will then go into the spinal cord, going up the dorsal column medial lemniscal pathway, and then synapsing all the way up in the brainstem in the medulla. A single neuron could be, if you're really tall, 2 meters in length. Really long neurons. 1 neuron. Anyway, it'll synapse onto that neuron in the medulla. This is all on the ipsilateral side of the body, right? So if this person's, say this person is facing us, so this is this person's right-hand side, it will be synapsing onto the right medulla. The neuron in the medulla decussates and projects to the thalamus, specifically the ventral posterior lateral nucleus of the thalamus. This nucleus then projects up to the cortex, where it connects to the somatosensory cortex, which we're talking about in just a moment. So this is 4 neurons in a series, where actually the 4th neuron is not drawn here, but it would be up here in the cortex. We would draw it like a little circle, and then it would have an axon connecting to other areas of cortex. Primary somatosensory cortex to secondary cortex. We'll get to that. So 4 neurons in a series, going all the way from the periphery up to the cortex. Keep in mind where the decussation is occurring. It is occurring here in the medulla. So the pathways, they decussate, coming from one side of the body, crosses to the other side of the brain. So it is, in this, like we said, this person's right-hand side, going up the right-hand side of the spinal cord, getting up to the midbrain, or to the brainstem, and then it would decussate and then go to the left-hand side, so the left-hand cortex would be perceiving the right-hand mechanosensation. So that's the transmission part. How does the information get up into the brain? Now we're going to spend a little bit of time talking about the perception side. It's pretty cool stuff. The somatosensation has a somatotopic map along somatosensory cortex. So all these pathways that are making their way up into the somatosensory cortex, they're actually organized to maintain information about where along the body these things are occurring. And the map is not organized in a super logical way. It doesn't look like a human body that's like mapped upon the brain, but it's a bunch of parts of the human body that are mapped upon the brain. And we refer to this map as the cortical homunculus that shows how the different parts of the body are represented within somatosensory cortex. The other thing that you should note is that you should see how there is distortion of the somatosensory homunculus, or the somatosensory map, of certain parts of the body in relation to other parts of the body. So things like the fingers, the hands, the lips, the mouth, these things are much larger and take up a much bigger part of somatosensory cortex than things that have fewer neurons and have much larger receptive fields, such as the arms, the legs, those kinds of things. They take up a relatively smaller portion, especially compared to skin surface area, relative to the hands and the mouth. So there's this distortion that allows, and this just has to be this way so that you can have a greater level of sensitivity to the information and process that information, so that you can manage all of that information about having the very small receptive field size at the tips of your fingers, whereas you don't need as much brain space dedicated to processing that information from, say, the upper arm or the thigh. So this is the primary somatosensory cortex that has this cortical homunculus. The primary somatosensory cortex, this is like the basic final stop for this this pathway in getting from the up into the brain. Okay, so the final stop of just the transmission of that initial information, and then from here things start to become processed and relayed to other brain areas so that it can be conceptualized and recognized. And this is another way for us to look at this overall cortical map. So this is going to be one of really the only parts of this entire course where I'm going to be referring to Brodmann's areas. I did mention that, you know, primary visual cortex or striate cortex, or V1, is also known as Brodmann's area 17. But this is the only part where, like, it actually makes a lot of sense to use Brodmann's areas. So somatosensory cortex is located in the postcentral gyrus of the parietal lobe. Okay, so we have the central gyrus, and it's located it's located caudally to that. Hence the postcentral gyrus. It's comprised of 4 subregions. We've got Brodmann's area 3A, Brodmann's area 3B, Brodmann's area 1, and Brodmann's area 2. Here's a color code for all of these different areas. And then from there, just like we have V1, so primary somatosensory cortex is known as S1, just as primary visual cortex is known as V1, we also have secondary somatosensory cortex, which is known as S2. So S1, S2. But in primary somatosensory cortex, we have these little subdivisions as well. And each subregion contains a somatotypic map of the body, with the foot and trunks and forelimbs that are on the medial side, and the foot right next to the genitalia, deep there inside the brain. I don't know how central it is to our thinking, I guess. I don't know. Joking. So you've got the map with the legs and the feet on the medial side, going all the way down, where then you have the trunk, the arm, the forearm, and then you have the hand, with this big representation of the hand that's more on the lateral side of the brain, on the outer surface of the parietal lobe. So then you have areas of the face, and then stuff inside the mouth, and that kind of thing. So, and each of these areas has this organization. It has exactly this organization. What you're looking at here in C is referred to as the homunculus. So this is what, if our bodies actually looked like the way our brain represents this information, this is what we would look like. Yeah, you would have these giant hands, big old lips, big old tongue, relatively small arms, small legs. This guy skipped leg day clearly. You know, he's got those squeezer things that just kind of like, you know, getting those giant hands all pumped up. Um, and of course, he's got the genitalia there too. So there's that. Yeah. So that's the homunculus. So the reason why we're focusing on these different Brodmann's areas is because 3B receives the majority of the projections. So 3B is the primary site of of the uh, of the dorsal column medial lemniscal pathway, and so that's going to receive all those projections, and then from there, 3B then has some processing of information that then is then sent to areas 1 and 2. As I mentioned earlier in this presentation, that animals like rodents, they have whiskers, hairs on the front of their face that they use for sensing their environment. This is actually a critical way, a critical modality that rodents use for perceiving the world. And since it is so important, the representation of the whiskers in their nervous system is actually quite large. Sort of like how the fovea in the visual system actually takes a large part of the visual cortex. The same is true for our hands, as we learned about with the human homunculus. But if we want to look at the mouse homunculus, they have very large representations of those most sensitive areas of their body, and it happens to be a big part of that is the whiskers. So the whiskers have a very stereotyped pattern. It's almost like a grid system, and we use number and letter coding to tell which whisker we're actually talking about. This number coding is actually represented within the brain as well. So we can identify which whisker that we're talking about here. In this case, we're talking about C2. This grid is represented in the cortex. It's laid out in a, not exactly like a perfect grid, exactly like the face, but it's pretty close. And all of those whiskers are there, and they're highly stereotyped, and they're highly represented. You can use various staining methods, and you can see the individual blobs within the brain, they're referred to as barrels, that represent the individual whiskers. And this is a picture of barrel cortex from a mouse cortex. And you can see well-defined barrels representing each whisker. The research in barrel cortex has actually revealed a ton of interesting information about how somatosensory cortex works, but also about experience-dependent plasticity, such as sensory adaptation when you remove a whisker, or when there are other experiments where you can stimulate a single whisker in order to enhance activity within that single barrel. And so you can induce all kinds of changes within a specific part of the cortex, leaving the rest of the cortex to serve as a control. So it's a really interesting, elegant system. The Olson lab is in fact doing some experiments using barrel cortex. They are very interested right now. They're doing projects related to understanding how BDNF, brain-derived neurotrophic factor, and the receptors for BDNF involving glia within the cortex, how that regulates the development and establishment of synapses within, and they're using barrel cortex as a model for that. Really interesting stuff. All right, so then we've got primary somatosensory cortex. We've got these 4 subregions, 3A and 3B. 3B receiving most of the input from the periphery, and then you have areas 1 and 2, which receives input from 3B. Those also then connect to secondary somatosensory cortex, which also has a map, but this map starts to get a little bit fuzzier. And then from there, it can project to other areas, such as the amygdala or the hippocampus. You will have projections from these areas going to premotor areas, which is right next to the primary somatosensory cortex. So you can use this somatosensory information to inform memory and fear information and help to contribute to motor information as well. Because like you have to know if you're going to respond to, say, being pushed and your limbs are sort of moving around, you need to know where your limbs are in order to have a reaction, and your body needs to know the position of the limbs in order to cause the muscles to fire in a certain way in order to have your limbs react, right? So all that information needs to be connecting and moving forward. But for as far as somatosensory cortex, that's essentially what we're going to be talking about from here. I do want to talk a little bit about somatotopic map plasticity, though. So this is some of the information that we do talk about in my neuroplasticity class. You know, I'm not going to be like trying to sell my class to you guys, although I guess I suppose I am a little bit. It's a super interesting class. You should really take it. But I won't cover everything that I talk about in my class in this class. But this is one that's particularly interesting, I think. It kind of shows the amazingness of this overall phenomenon, and it, you know, the brain, and I want you to have an appreciation as well for the fact that though the brain is quite set in stone in many ways, that it's not completely set in stone, and that some change occurs over time depending upon conditions. So we saw the mouse cortex with the big barrels. We saw the human cortex. There's been a lot of work on somatosensory cortex in monkeys as well, and they actually have a somatotopic map that's quite similar to the human somatotopic map, with the legs next to the arms and next to the forearm, and then you have the hand and then the face. And we can work on this. There have been times in mouse colonies when, monkey colonies, when a monkey may have fought with another monkey, and then the hand might get infected, and then unfortunately they will amputate that monkey's hand. It's something that would occasionally happen in some of these colonies. But John Cass, in his lab, he had was interested in the somatosensory cortex and how this works, and had taken advantage of some of these monkeys to see how has their overall nervous system changed. So if we wanted to see the projections going up through the spinal cord, there are a few ways that we can look at it, right? We can do the cross section, in which we saw before, where you can see the different parts of the spinal cord, or you can lay it flat and then cut it flat, just sort of like you're cutting a baguette to make a sub sandwich, right? That's another way that you could cut the spinal cord. So that would be like a transverse cut along the spinal cord. And so they had done this in this experiment, and what they had done is they had injected the arms with a compound known as horseradish peroxidase, which fills and labels neurons. And then they wanted to see where are the projections in the spinal cord from these arms. So this is taken from a monkey who had been amputated many years before, and when you do these injections in a normal monkey with with both hands present, and you inject these parts of the arm, you can see horseradish peroxidase traveling up through the spinal cord. So these processes going from the primary afferents that would then connect to the dorsal column nuclei and the medulla. We're not quite at that stage yet. We're just looking in the spinal cord. This is where these afferents would be coming in. So the dorsal root ganglia would be over here and be connected over here. So it would be this particular dermatome that would be coming in through here. And so that's what it would be normally, right? If in these monkeys that were amputated, when they do these injections, they see something very different. So instead, what they see is that when they inject along the arm, not only do they see a bunch of horseradish peroxidase in the parts of the spinal cord that would receive input normally from the arm, but they also see a bunch of projections coming in from where would be the hand. So in a normal monkey, if you were to inject horseradish peroxidase in a bunch of places within the hand, you would see a bunch of black here, but you don't, because they only injected the arm. But when you inject an amputee monkey, they don't have a hand to inject, and instead you get a ton of input coming in to the arm. So that's interesting. What's going on there? So this has been an issue for people, and presumably for monkeys as well, leading to a phenomenon that's known as the phantom limb syndrome. And so what's happening then is that you have all these afferents going into that part of the spinal cord, and then eventually making its way up into the brain. And what's happening is that these areas that would have normally made up the hand, so remember you've got the arm, we're looking at area 3B here, you got the arm up here, you got the face here. Normally these would be areas that would be innervated by the hand. But instead, what they had done is that they were recording from sites within the brain, and in this monkey with the amputation, and they were touching parts of the stump and looking to see which parts of the stump were activating those neurons. And you would think, okay, well there should be no activity because there's no hand. But instead, this whole area has now been filled in with sensitivity to the stump. And presumably the monkey is perceiving this, the brain sees this as the hand, and it might even perceive it as it's still having a hand. And this is why many amputees have what is called this phantom limb syndrome, where they, even though the hand is not there, their brain is telling them that they feel like they actually have a hand still. And so in my neuroplasticity class, we talk about some of the underlying mechanisms of why this occurs, that there's some new axons that grow in, and how 3B then acts on areas 1 and 2. That's part of the reason why we focus on these little subdivisions of primary somatosensory cortex. Yeah, so it's a really interesting phenomenon, and it turns out that it's because there's preservation of these brain. So even crazier is that the hand area also responds to touches along the face. So this is another study where what they had found is that the hand, so this is the deafferented cortex here, and if you touch along the face, because the face is right next to where the hand part is, and the hand is missing, these dots are dots that were sensitive to face touches now. So the face has now expanded and now pushed into this part of the cortex that was normally innervated by the hand and the arm. So you get massive cortical reorganization that occurs when a limb is missing. And it's because these areas, they're still there and they're hungry for input and they need input, so you'll get this reorganization that occurs. This is something that also happens in there's a famous cognitive neuroscientist and neuroimaging specialist. His name is V.S. Ramachandran. He is at UC San Diego, and he's done a fair amount of work on this whole phantom limb syndrome in humans. This is a very interesting video. I will put a link in the description below. I'll try to put a card up on the YouTube video here. If you have the PowerPoint file, you can just click on the link here. I suggest you do that now. I will pause and wait for you to do that. Great. Welcome back. So, we've learned a little bit about what's happening there. Over time, what is happening when these areas are completely deafferented. So if you've lost the arm, you have the face and the arm trying to fill in this space. So the arm is going to increase its sensitivity, and it'll feel like you still have a hand there, and many times these people still feel like, I still have a hand there, and I can feel like my hand is at the stump itself. But even then, you will also have the face starting to overtake some of these things, and you'll get cross modalities, cross sensitivity of these sensory inputs, where from touching on a single place, it'll feel like it's touching on two different things, one of which doesn't exist, but the person still feels like it exists. Crazy stuff. Okay, so to wrap up, here are the different key questions. What is somatosensation and proprioception? What are the key mechanoreceptors that process tactile information? What is the anatomy of the mechanoreceptor neurons, and where are their cell bodies located? Dorsal root ganglia. What are receptive fields, especially as far as we refer to them as somatosensation, and how do they vary across the body in size and sensitivity? What are the key receptors that are involved in proprioception and how do they work? What is the anatomy and connectivity and mechanosensation from receptor neurons in the cortex? What is somatosensory cortex? How is it organized? Where is it located? What is the homunculus? What does that look like in humans and mice? And then how does amputation affect the somatosensory cortex? All important questions. So that's it for this lecture. Looking forward to seeing you in the next.